see the presentation? But we can hear you.
Oh, okay. Can you hear me now?
A little bit. It's a little bit low.
Okay. How about now?
Now it's better. Try to talk.
Hello?
Hello. It's for me. It's a low. I don't hear very well. I don't know the other people, but okay. You are not in the presentation mode. Okay, perfect. You can slideshow. Yeah. Yes, it's yeah, on the back. Perfect. So you are in presentation. Okay, I hope we can hear you. The show is yours. Abiola, welcome.
Thank you for
We can
having me.
hear very well. Can you?
How about now?
Well, we'll see. Let's see. Try to speak louder as possible. I hear.
Okay. I will try to raise my voice now. How about now?
Better. Try to speak because I hear.
All right. Thank you very much. Thank you for having me. My name is Abiola Abdülrahman Ayanlara from Shuzhou Medical University in Jiangsu Province, China. And I will be presenting a part of our work titled Doublecortin promotes glioblastoma, glutamine metabolism via oxidative phosphorylation and regulates homeostasis in favor of glioblastoma cell development. So glioblastoma is also known as GBM, and it is an aggressive type of brain tumor, and it is very difficult disease to treat. It is characterized with invasive abilities that fuse the infiltration, uncontrolled proliferation, resistance to therapy, and reinitiation after surgical resection. And the median survival rate of GBM is less than a year, and there is currently no cure for glioblastoma. So we know tumors reprogram pathways of nutrient acquisition and metabolism to meet the bioenergetic and biosynthetic and redox demands of malignant cells. And these reprogrammed activities are now recognized as hallmarks of cancer. And tumor cells harbor genetic alterations that promote continuous and elevated production of ROS, reactive oxygen species. And whereas such oxidative stress conditions would be very harmful to normal cells. So they facilitate tumor growth in multiple ways by causing DNA damage and genomic instability, and ultimately by reprogramming the cancer metabolism. So alterations observed in cancers involve the deregulation of amino acid management, and it is well established that amino acid glutamine is among the most highly consumed nutrients in cancer cells, and the biosynthetic pathways that metabolize it are required in various cancer subtypes and are object of current efforts to target cancer metabolism. However, specific mechanisms by which alterations of amino acid management promotes cellular transformation are mostly elusive. So it is therefore imperative for fuller mechanistic understanding of these processes, and it will be important for efforts to exploit such alterations for cancer therapy. So in our lab, we've been working on doublecortin. Doublecortin is also referred to as DCX, and it is a neuronal microtubule associated protein. It was first described with regards to its mutation in human X-linked leukoencephaly and the double cortex syndrome, also known as subcortical banded heterotopia. And these are two neurodevelopmental disorders associated with the abnormal migration of cerebral cortical neurons. And DCX is crucial for neuronal migration and the cytoskeletal organization by stabilizing microtubules. And in fact, it is a gold standard marker of adult neurogenesis. DCX is expressed in gliomas, irrespective of tumor grade, type, and position. And in our most recent research article, we have reported analyzed data sets from from Oncomine database, showing significant expression of DCX in different glioma datasets compared with non-neoplastic tissues, and simultaneous expression of DCX with semaphorin 3B and osteonectin translates to poor prognosis in glioblastoma. So the crucial roles of DCX in adult neurogenesis shows newly formed neurons from their origin express DCX. So neurons are formed in the subventricular zone, subgranular zone, and the dentate gyrus, moving through the rostral migratory stream, to their destination. The olfactory bulb are highly expressive of doublecortin. So because of this, because of the functions of DCX in adult neurogenesis, it is imperative to study the reason why DCX is expressed in glioblastoma. And our previous works has revealed translocation of DCX from the cytoplasm of glioma cells to the nuclear compartment, where we identified the nuclear localization signals, and we reported that this translocation of DCX from the cytoplasm of the glioma cells to the nucleus will further improve the development of glioblastoma, because DCX is initially regarded as a cytoplasmic protein. So our previous work also revealed that the translocation of DCX would improve migrative and invasive capabilities of these cells, of these cells expressing IDCX expression, and also it would improve tumor mass when in our immunocompromised mouse model. So we also reported that at the edge of the invasive glioma tissues, the cells at the edge are nuclear DCX positive, and DCX in glioblastoma is a new one. However, our findings show a positive involvement in metabolic activities in GBM cells. So to further the research, the functional properties, the research on the functional properties of DCX in gliomas, we carry out in vitro experiments using the P53 mutant and P53 in all glioma cell lines, and we also use the hepatocarcinoma cell lines. We also use the hepatocarcinoma cell lines. However, in this presentation, I would only be presenting on the experiment carried out on glioma cells. So cells targeted for IDCS expression labeled SG0, while those targeted for DCS downregulation are labeled SG4. And to start with, we confirmed the effect of IDCX expression on some of those cells, and we observed higher growth confluence in the SG0 cells after 2 days, while the SG4 cells, on the other hand, exhibited stunted growth and irregular shape. So we analyzed cell checkpoints using the flow cytometer, and we observed that. Observed significant improved G2M phase in SG0 cells, and this is the phase where the cell replenishes its energy, stores, and synthesizes proteins necessary for chromosomal manipulation. Also, cell counting proliferation experiment showed significant proliferation rate in SG0 cells, while growth rate in SG4 cells were sluggish. And in context, for the SG4 to grow to 70% confluence, it would take up to at least 7 days to 8 days for a process that should only last about 32 to 48 hours. And this indicates that DCS is essential for glioma cell growth. Then we carried out whole genome sequencing experiment on C6 astrogloma cells compared with the CRISPR-Cas9 targeted overexpression cells with RNA-seq experiment. And our GO analysis of the RNA-seq results showed implicated differentially expressed genes could be involved in cellular growth, biological regulation, metabolic processes, and development. And furthermore, our KGG enrichment analysis indicated cellular signaling, including transport, catabolism, protein synthesis, drug resistance, amino acid, lipid, and carbohydrate metabolism, and metabolism of other amino acid and energy metabolism. And it also shows transcriptional manipulation in cancer, HIF1 signaling, as well as nitrogen metabolism. And based on the analysis and observation of these enriched genes, we selected 4 differentially expressed genes, including asparagine synthetase, activating transcription factor 5, SLC7A11, and glutathione S transferase. For this presentation, we will focus mainly on asparagine synthetase. So while glutamine can be generated biosynthetically in vitro, most cancer cell lines engage exclusively on the conversion of glutamine to glutamate. And by the contribution of nitrogen from the glutamine side chain amine, either to de novo nucleotide biosynthesis or through the release of amine as free ammonia by the action of mitochondrial glutaminase. And the contribution of glutamine to amino acid biosynthesis establishes it as a key ingredient to the protein translation needs of cancer cell. And this will help in inhibiting autophagy. So in our RNA-seq analysis, we observed the enrichment of glycolytic pathway as GAPDH as GAPDH is shown to be highly expressed in this analysis. So we checked the mRNA levels to determine the enzymes catalyzing the central carbon intermediates of the glycolytic pathways, and we observed that all glycolytic enzyme probed were highly enriched in DCX overexpressed cells. And most importantly, the results here corroborate that derived in the RNA-seq showing GAPDH enrichment in SG0 cells. However, the rate-limiting enzyme, the PFKFB3 enzyme showed lower expression, indicating that although there is high level of glycolysis going on in DCX overexpressed cells, only two molecules of ATPs are generated in the second half, and this indicates a shift in energy requirements from TCA cycle, from glycolysis towards TCA cycle. So we later checked the G6PC to confirm a shift towards gluconeogenesis, and the GLS2 to further confirm glutamine regulation. And we could observe that there is a higher expression of G6PC and also higher expression of the GLS2. Also, further analysis of RNA-seq showed the enrichment of TCA cycle, as it indicates enriched oxaloglutarate dehydrogenase complex, which produces the Can you hear me?
Yes, thank you so much. So what happened?
I don't know. Is my screen still on?
No, is not. No, you are not sharing the screen. Yeah, maybe that was the problem. Yeah.
Okay. I was here, right?
Yeah. Yes, you were here, the previous one. Yes, this one.
This one?
Yeah.
Okay, thank you.
No.
I think something is wrong with my internet. All right. So we verified the expression of the TCA cycle intermediate, and our results indicate that DCX enriched TCA activities when compared with the control groups, and this indicates complete production of ATP molecules in the TCA cycle, as well as NADH, FADH, and the CO2, as well as hydrogen molecules. So because of SCO2's function in hypoxic tumor and the P53 state of our cells, we check the SCO2 expression to see if there is a recapitulation in the metabolic switch towards glycolysis, and we could see that SCO2 expression was lower when compared with our control group, and we further detected mRNA of tiger, and and we further detected the mRNA of tiger, which is highly expressed, and we checked the GSH level to confirm that GSH, and we confirmed that GSH is high in DCX cells, which indicates there is a protection against ROS-induced apoptosis. So we confirmed the presence of ROS in these cells by checking for extracellular ROS level with DHE staining. And after this confirmation, we also confirmed if our cells can, if DCX can protect our cells against stress. And we cultured the cells in different nutrient conditions, in no glutamine condition, low glucose condition, as well as no glucose condition, supplemented with galactose instead of glucose. And we could observe that DCX stabilizes the cells better in cell condition, in these cell conditions than in the control groups, while in the downregulation group, downregulation of DCX group, we observe significant loss. It significantly loses its proliferative effect capabilities in nutrient depleted condition, and activates apoptosis in mass in no glucose medium. So we tried to confirm what happens to the differentially expressed genes in these culture conditions, and we could confirm that although asparagine synthase expression decreases drastically in the no glutamine group, the glutamine deficient culture medium, the protein translation complex also showed similar results. However, there is high expression of this expression of EIF2 alpha in the no glucose group. And our results in the beta complex of this particular enzyme shows depletion. So to probe the importance of the stability of the alpha complex of protein translation, we utilized the ultra-high pressure liquid chromatography mass spectrometry to probe amino acid levels in our cells compared with high DCX cells and DCX knockdown cell in normal and no glutamine condition, as well as in different culture condition per se. And as predicted, we found high expression of DCX in the high expression. We found that the high expression of a common amino acid in DCX overexpressed cells, and similar results were observed in cells cultured in glutamine deficient medium. And this indicates abundance of intracellular amino acid in high DCX cells, including glutamine, glutamate, and asparagine. Then we inhibited asparagine synthase with L-asparaginase and observed similar results as detected in the glutamine deficient medium. Sorry. Can you see my screen?
No, no. Now we are not sharing.
Apologies. Can you see my screen now?
No.
How about now?
Now, yes, we can see the screen.
Apologies. Okay, so we
almost 5 minutes.
Oh, sorry. Okay, so similar results were observed in the when we inhibited the asparagine synthase enzyme with L-asparaginase and observed we performed a semi-quantitative central carbon metabolomics experiment to determine the level of the metabolic intermediates using normal growth conditions, no glutamine, low glucose, and no glucose. And we observed the enriched TCA cycle intermediates like fumarate, the malate, GTP, CMP. And this further suggests that metabolic activities in the CX overexpressed cells via the mitochondria. So we checked the mitochondrial morphology for via the transmission electron microscope, and we observe shrunken and swollen mitochondria in the DCX down-regulated cells. And when glutamine was withdrawn from this from the medium, we could observe a change in the mitochondrial shape of the control group. And when DCX when glucose was withdrawn, we observed autophagy in our SG0 cells, while lysis and apoptosis was observed in the control cells. So with the previous results pointing towards oxidative phosphorylation and RNA-seq analysis indicating high oxidative phosphorylation in the cells, we finally observed the oxidative phosphorylation in the cells by checking the substrate uptake and the OCR and ECR level using Agilent's C-horse metabolic stress test. And we observed OCR OCR and ECR level in IDCX cells, while the DCX knockdown cells showed less OCR and ECR, as well as substrate uptake, and when cultured in glutamine deficient medium condition. However, in the glucose deficient condition, medium, the SG4 cells showed a shift away from glycolysis as they have higher glutamine uptake level and OCR. So for glucose pyruvate stress test, we observed similar results as SG0 cell had higher uptake even when glutamine has been depleted. And also similar results were observed in fatty acid oxidation stress test, indicating that DCX promotes glioblastoma cell proliferation via oxidative phosphorylation. So we also checked the NADH levels in the cells as we confirmed that SG0 cells exhibited higher levels of NADH levels in normal culture condition and in glutamine deficient culture condition, while in low glucose medium, we observed higher NADH levels, and this may be attributed to the sodium pyruvate levels in this culture medium. So to further confirm the metabolite interaction with DCX, we carried out mass spectrometry for protein-to-protein interaction in control cells compared with the DCX overexpressed cells, and the KGG analysis showed enriched interactive proteins are involved in oxidative phosphorylation, metabolism, glutathione metabolism, fatty acid, and amino acid metabolism. And DCX directly binds to energy generation molecules, as well as amino acid transporters and central carbon metabolites. And DCX also directly binds to molecules involved in protein synthesis. And we also could observe EIF family members binding directly to DCX. And finally, we are currently doing in vivo experiments in syngenic and congenic model. And we are also doing experiments on the other differentially expressed genes we mentioned earlier to confirm DCX metabolic activities, promote drug resistance and proliferation. And we are using the combination of the inhibitors, erastine, sorafenib, and coserine, as well as L-asparaginase and cycloheximide in the congenic and the syngenic model, mice model. And we have also identified and finished epigenetic studies on the relationship between asparagine synthesis and transcription factors in DCX overexpressed cells. And this gave us the relationship between the enzymatic activation of glutamine metabolism, as well as the homeostasis via the exchange of glutamate and amino acid synthesis. So in conclusion, we concluded that DCX promotes glutamine metabolism in glioblastoma cells, and it is essential for metabolic activities in DCX. And glutamine is essential for metabolic activities in DCX cells, but relies on glucose for survival. Also, oxidative phosphorylation is a major source of energy for glioblastoma cells expressing IDCX. And DCX is also a promising target in glioblastoma therapy. So I would like to give special thanks to the members of my group, most especially Chembo, Ikra Nadim, Xiaolian, and also the new PhD student in our lab, Kanwore Kominin, as well as Peniel Afayo Kambe. And I would like to give a special thanks to my professor, Professor Gao Dienshui, for entrusting me with a team of my own, and also the members of the Public Experimental Research Laboratory of the Department of Neurobiology and Anatomy. I'll also like to give a special thanks to the National Natural Science Foundation for funding this project, as well as PAPD, Jiangsu Province, Xinlan project. And I would like to give special thanks to the organizers of this conference, most especially Professor Hira, for always including us in the scientific discussion from their laboratory. So on the last note, I would like to let you, for those of us who are doing the metabolic experiments in this particular session, if you have a postdoctoral position, you could contact me from this, via this email at the bottom of the screen here. So thank you very much for having me, and thank you all for listening.
So thank you so much for this very nice presentation. Thanks a lot. So there are, let me check if there are any questions in the chat. I have maybe a question. I didn't get one point. How long did you take the cells with the different conditions, low glucose or no glutamine, no glucose? How long did the cell were exposed?
So usually the culture condition for the cells in the normal growth medium is 2 days before passaging, before the subculture. So when we withdraw the glutamine or the glucose, we normally withdraw it and then let it go through the normal culture condition, then passage maybe twice or 3 times before we carry out the experiments there. However, for the no glucose group, the one which is supplemented with galactose, it cannot undergo that subculture period. So most time we just culture them for 2 days before we can use them for subsequent experiments.
I see. And what about the heterogeneity of the cells? Did you analyze that? Maybe I think there are many different clones in this cell, and did you take care of the different? Maybe some clones are more affected than others.
You mean different type of cells were used actually?
Yeah. I mean, you didn't take care of the heterogeneity of each type of cells. I mean, taking into account one cell, no, there are some subclones.
Yes.
Usually.
We used the P53 as a decisive selection for the particular cells we use. So some of them are P53 mutants and some of them are P53 null. So we also use the hepatocarcinoma cells for our experiment because they are expressive of double protein.
I see. Thank you.
Also, so the results were not shown in this particular presentation.
Thank you. So there are more questions? No. So I